If a plant has enough water, minerals and energy, it will grow, right? Well, sort of. But there is more to it, like why do plants bend towards the light and not just grow straight? And why is it that the stem grows up but the roots grow down? It isn't as if the plant has eyes to tell it where the sun is. You should already know about tropisms, so phototropism and geotropism. But if you have forgotten, watch our video called Plant Hormones, Tropisms first. But how does it actually do this? Having been made in the tips of the stems and roots, auxin is moved in solution by diffusion to older parts of the plant. In the stem, the auxin causes the cells to change in elasticity. More elastic cells absorb more water and can grow longer. Strangely though, stems and roots respond differently to high concentrations of auxin. Whilst the stem cells grow more, the root cells actually grow less. So auxins make plants grow. But why do they bend towards the light? How do they know to do this when they don't have eyes? The bending happens because the light hits on one side more and breaks down the auxins on that side of the stem. So then growth slows down on the light side. The faster growth on the dark side causes the shoots and leaves to turn towards the light, which is ideal for the plant for photosynthesis. Auxin is produced in the tips of growing shoots. If the tips are cut off, then no auxin can be produced and so no plant growth. If the tips are covered whilst auxin is still produced, light cannot break it down and so phototropism cannot occur. The plant just grows straight up and does not bend towards the light. Pretty straightforward so far. Now let's switch it up a bit. Auxins have the opposite effect on root cells. In roots, auxins cause less growth. The shaded side of roots contain more auxin, and so they grow less. This enables the light side of the roots to grow more and bend away from the light. And if that wasn't weird enough, we have the opposite happening with auxins to gravity too. In a horizontal root, the bottom side contains more auxin and grows less. So the root bends downwards in the direction of gravity. So positive geotropism. But of course, the stem responds differently. In a horizontal stem, again, the bottom side contains more auxins because it is less directly hit by sunlight. But because auxins cause growth in stems, the bottom side grows more, causing the stem to bend upwards, against the direction of gravity. So, negative geotropism. But I'm giving auxins too much credit. They don't work alone. They have a partner in crime. Cytokinins. You don't need to know anything about these hormones, other than the fact that they work alongside auxins. There is another plant hormone, however, that you do need to be aware of. Gibberellins. Once a seed germinates, the roots and shoots start to grow. But for this, the seed needs energy. Luckily, the seed releases a hormone called gibberellin, which causes the starch in the seed to turn into sugars and provides the seed with energy to grow. As well as causing shoot growth, gibberellins can also stimulate flowering and the production of fruit in some plants, and they also work with auxins to cause stem elongation.
Plant hormones, also known as plant growth factors, are chemical substances that accelerate, inhibit, or otherwise affect growth. This animation explores the action of indole-3-acetic acid, IAA, the most common form of auxin, and looks at how we discovered what auxins do. The auxin IAA is produced at the apex of the stem. Under unilateral light, shining on one side, IAA is redistributed to the shaded side, creating a gradient of the hormone across the stem. IAA travels between cells by diffusion. IAA activates proton pumps, which lowers the pH. This activates enzymes, expansins, to break bonds in the cell wall. The loosening of the cell wall allows water to enter the cell and turgor pressure increases. Cells on the shaded side become elongated and cause the shoot tips to bend towards the light. But what is the experimental evidence for this? Darwin investigated using oat coleoptils. He found that there is no bending response to light when the coleoptil tip is removed. This means that either the tip is the origin of the messenger, or it must be stimulated by light. To investigate further, Darwin covered the tip to prevent light from reaching it. The lack of bending response confirmed that the tip must be stimulated. When light was shone on the tip but the lower regions were covered, the coleoptile did bend. This showed that only the tip needs to be stimulated. Peter Boysen-Jensen used a thin flake of the mineral mica, which blocks chemical movement. When it was inserted in the illuminated side, bending occurred normally. But when the mica was inserted in the shaded side, there was no bending, showing that the message to bend travels down the shaded side. Because the mica doesn't block electrical signals, this also suggested that the message was chemical. Arpad-Pal's experiment, replacing a segment of the tip off-center, increased understanding of how the hormone is distributed. Even in the dark, the coleoptile bent towards the side without the tip. Fritz Wendt removed the coleoptile tip and incubated it on a block of agar. The agar was then placed on the coleoptile off-center in darkness. Hormone that the agar had absorbed travelled down the side of the shoot, causing a response. The amount of bending is proportional to the amount of hormone in the agar. Winslow Briggs inserted a very thin glass plate vertically through the tip. The glass does not prevent light transmission, but there was no bending. Auxin cannot travel through glass, and its concentrations remained equal on either side. When the glass plate was inserted below the tip, auxin moved across to establish a hormone gradient. When the tip was examined, 70% of auxin was found on the shaded side.
Cytokinesis in plant cells is different from that in animal cells. In early telophase in plants, vesicles carrying cell wall precursors from the Golgi apparatus associate with remnants of the spindle microtubules and accumulate at the former side of the metaphase plate. These vesicles then fuse to form a large membrane-enclosed disc-like structure, and their polysaccharide contents assemble to form the matrix of a new cell wall called a cell plate. The cell plate expands outward, perpendicular to the spindle, until it reaches the plasma membrane. The membrane surrounding the cell then fuses with the parental plasma membrane, dividing the cell in two. Connections called plasmodesmata between the daughter cells are formed as a result of incomplete vesicle fusion during cytokinesis.